podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience from Harvard University, where I focused on neuroendocrinology and the neuroscience of behavior. And before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to parse and translate the information guests share on the show. And in addition to the podcast, I write long-form content and send out a free weekly newsletter at the Mind and Matter Substack at mindandmatter.substack.com. I also offer private, science-based health consulting, where we work one-on-one and apply the same rigorous, evidence-first approach you hear on the podcast, but personalized. We can obtain and analyze your biomarkers, work on diet and behavior strategies, review supplements and drugs that you might be taking, or anything else related to general metabolic health. So if you'd like more tailored guidance on your personal health, head to nickjacomis.com and fill out a short form or book time with me there. A great way to support my content in all of its forms is simply to visit mindandmatter.substack.com. You'll find a link to that in the episode description, and you'll also find a link to my current affiliate partners through my support page. These are products that I use myself that relate to physical, metabolic, or mental health. For example, one product I use every day is the AquaTrue water filter, which removes heavy metals, microbes, endocrine disruptors, you name it, so that you have higher quality drinking water. All of those links for health consulting, for my affiliate partners, and the Substack page are all in the episode description. So thank you very much for listening, and don't forget to check out mindandmatter.substack.com for all of my content. Dr. William Banks, thank you very much for joining me.
Good to be here, Nick. Thank you for inviting me.
Can you just tell everyone a little bit about who you are and what your background is as a scientist?
Sure. I grew up in Southeast Missouri and country boy, and I fell in love with nature and biology. And I admit I'm one of those geeky kids that set up a lab in my bedroom. And you can buy all kinds of chemicals then you can't buy now. So I set up a microscopy lab of sorts. Really fell in love with biology and medicine as an undergraduate, and then was lucky enough to go to med school, but decided that science was really what I wanted to do. And so after med school, I did internship and residency and that kind of thing under the auspices of two really great guys, Abba Kasten and also Andrew Shalley. And by this time, I was interested in how the brain and the body communicated. But I almost went into the vagus, but then I got interested in humoral communication. And just about this time, there were a lot of papers that were coming out that all of the peptides that had been found in the gastrointestinal tract were now being found in the brain. For example, cholecystokinin. And all the peptides that had been first found in the brain, like somatostatin, were now being found in the gut. And some of this work came out of a New Orleans lab with Abba Kasten and Andrew Shalley. And Andrew Shalley, in fact, had just won the Nobel Prize for the discovery of the hypothalamic hormones. And Abba was also interested, and that obviously connected the brain to the body in that direction by controlling the pituitary. And Abba was interested, did it go the other way as well? Because he would inject these hormones peripherally and find brain effects even in animals that didn't have a pituitary. So there was some cross communication there, and so we wound up being interested in the same problem. And the problem is that there was the blood-brain barrier, and everyone said peptides could not get across the blood-brain barrier. If they could, then that would be a way that you could get signaling from the blood into the brain or the brain into the blood, and so have brain-body communication. But people said, no, they're too big. I have no idea why this was such a dogma against that particular topic, but it really was, and still is to some degree in some circles. So we set it up as sort of a tautology, so that either way the answer would be important. And the idea was that if peptides could normally cross the blood-brain barrier, then that could be a way that the brain and the body normally communicate. But if they didn't, then when the blood-brain barrier was disrupted in various diseases, then maybe they could leak across, and that would be a way of brain-body communication during disease states and explain some of the pathology. So with that, we endeavored to determine, do peptides cross? We started out with a small peptide known as Peptide, which, again, this is back in the 80s. And it had the virtue of we could do various tricks and determine, does the full-length 9 amino acid peptide really get into brain? And does it do anything when it gets there? And we answered the questions. Both were yes. And then we did some enkephalin work and some other things. So after about 10 years of slogging away, we were able to say, yes, peptides can cross the blood-brain barrier, and they can do something significant once they get into the brain. And then once you accept that premise, that there's a humoral connection between the body and the tissues mediated by substances, circulating substances, you can study anything that you're interested in. You know, there's obesity. How do viruses get in? How do they affect the brain? Sleep, almost appetite, anything that you can think of that involves peptides in the periphery that have functions, have effects in the brain. You can study all kinds of things. And so that's sort of what I've been trying to do for the last 45 years.
Yeah. So brain-body communication, obviously, we're one integrated unit, the body is. The brain and the body have to communicate bidirectionally. One channel that can happen through is things like cranial nerves, like the vagus nerve.
Exactly.
And,
yes.
So nerves that come down from the brain and touch and directly interact with our organs and the rest of the body. Another channel that we knew about, a long time ago when you were getting started was the pituitary. So the brain, places like the hypothalamus, the pituitary gland, can secrete substances such as peptides that get into the blood and then go elsewhere. And so naturally, the question arose, it sounded like, well, does it go in the other direction? Can things get from the body and the periphery into the brain via the bloodstream? And, that brings us to the topic of the blood-brain barrier. So some people, I guess, thought, no, there's probably nothing coming from the periphery into the brain because that barrier is a barrier and it doesn't really let much of anything through. And of course, your whole career and some of the things we'll talk about, it makes the answer much more interesting and complex and dynamic than that. But I want to give people some background on the basics as we understand it today, before we get into some of the other work that you've done that's been fascinating over the years. From today's perspective and your understanding and everything that we've learned over the years, let's just start basic. What is the blood-brain barrier? And can you just give people a very high-level sense for its general structure?
Yeah, I think there's two common definitions, a more generalized one and a very specific one. The more generalized one, I think you could say there's a blood-brain barriers, and that these consist of all the processes you might think about that prevent circulating substances in the blood from getting into the brain. And then the more specific one, blood-brain barrier, is often used to distinguish it from the blood cerebrospinal fluid barrier or the choroid plexus. And in that case, the blood-brain barrier is essentially the capillary bed of the brain. And most people would include the immediate arterioles and venules on either side. And as your audience knows, I'm sure very well, that in most tissues, the capillary is a little bit leaky. And it's by that leakage that those tissues get all of the nutrients that they need, and that begins a lymphatic system that clears out toxins and all this kind of stuff. But in the capillary bed of the brain, there is no leakage. There is no production of an ultrafiltrate, a nourishing ultrafiltrate. And that's great because that keeps all the circulating riffraff out of the brain and makes it a pristine shirt sleeve environment to do whatever it wants to do. But on the other hand, it creates a problem. If there's no nourishing ultrafiltrate being produced, how does the brain get all the vitamins and nutrients that it needs? How does it get all the glucose it needs? It's the most metabolically active tissue in the body, 10 times more glucose being used per gram than almost any other tissue is what's commonly cited. And the answer then, interestingly, are the blood-brain barriers, the choroid plexus, the vascular blood-brain barrier, And some of the others, like at the circumventricular organs, there's a tanacitic barrier there, and there's a couple of other barriers that are very commonly listed now too. But the major one, and the one that most work has been done on, is the blood-brain barrier. And I often have to apologize when I'm giving talks that I don't really know what's going on at the choroid plexus. I just had to focus someplace in in my career, and so I've sort of ignored the choroid plexus. Although I think that many of the processes that have been found at the blood-brain barrier, particularly at the principal level, are also occurring there. And in some future reincarnation, maybe I can come back or some other great scientists can take on and compare and contrast these two barriers. Yeah. That was very popular in the 70s, but no one's really doing that right now, I think.
Well, before we dig into the details on the blood-brain barrier proper in just a moment, let's just give people a sense for some of the things that you just mentioned. Starting with the choroid plexus, what exactly is that on a very basic level?
So if you think about your brain, there's a hollow area inside the brain where there's a lot of CSF. And there's little bags of tissue, the choroid plexus, that produces the cerebrospinal fluid that it bathes the outer rim of the brain, the interior, and goes down to the spinal cord. And but it's not just producing cerebrospinal fluid. It's also dictating what the contents of that cerebrospinal fluid is. And in that way is delivering various substances or allowing various substances to cross.
Mm-hmm. So the choroid plexus is the sort of the barrier that involves the cerebrospinal fluid as opposed to the blood-brain barrier, which is the blood vessels carrying literal blood.
Yes, exactly.
Got it. And then you said circumventricular organs. That might be something we circle back to. What are those?
Imagine the blood vessels going through and the capillaries are there. And for a very short distance, those capillaries decide to have no barrier or a lower barrier. So those vessels become leaky. And there's 7 of those in the mammalian brain. There's all around the ventricles of the brain or near the ventricles, hence the name circumventricular, as near the ventricles. However, in most cases, they are delimited by tanocytes. So they're true pieces of the brain, they're true pieces of the central nervous system, but they have 2 different modifications. One, that their vessels are leaky, and 2, that those leaky areas are delimited by tanocytes, by a tanacitic barrier. So the things that leak into the into their interstitial fluid don't necessarily very easily get into the surrounding interstitial fluid of the adjacent brain. What does that mean? It means you've got these little pockets where the central nervous system changes the rules, and they're all of a sudden intimately exposed to what's going on in the blood. And it's interesting that many of these are involved in sort of emergency reactions, like blood pressure, or if you swallow a poison, these are the emetic centers and things like that. I'm sure they do a lot of more things, and the CVOs are being studied by a small group of very talented people. So I think that's an area that's going to really grow a lot over the next decades.
So basically, there's a blood-brain barrier that is regulating and limiting what can go from the blood into the brain. But these circumventricular organs are special or exceptional parts of the brain where that barrier is less strong, and the brain, specific parts of the brain, can more easily sample things that are in the bloodstream.
Right. So you see that things get very interactive and complicated and sort of a yin and a yang, and for every rule there's an exception, and for every exception to the rule there's an exception to that rule. And this makes studying the blood-brain barrier really intriguing, because all of this complexity is fine-honed. It's beautifully put together. It's not sort of slip shot or something like that. So if you can wrap your mind around it, you begin to see all these opportunities by which the brain can play one rule off against the other, and get very, very sophisticated in how it can have its cake and eat it too, and how it can respond elegantly to communication. And all of these are also opportunities for drug delivery to the brain. We probably will talk about that later, but the blood-brain barrier is the biggest obstacle to developing drugs for the central nervous system.
Yeah, yeah.
You know,
So a central conundrum here from the body's perspective is how do we, on the one hand, protect the brain and not just let anything in the blood get into the brain, which could be very problematic. But on the other hand, if we build a barrier that prevents bad stuff that we don't want to get into the brain from getting into the brain, how do we allow nutrients and all of the other stuff that we actually want to supply to the brain? How do we allow them to actually get through? And, we're going to dig into some of that. But if we just think very high level to start with here, and you said something interesting. There's general rules here, and then there's exceptions to rules. So we're talking about tendencies. There's going to be lots of exceptions. There's nothing that's like ironclad here necessarily. But as a general rule, how do the basic properties of substances relate to how easily they can get through the blood-brain barrier? Their general size, their charge, their water solubility. How do these things start to factor into whether or not something is more or less likely to pass that barrier?
Right. There are, again, general rules for which substances can get in, and there's different classes depending on what they use. And the big two are saturable, those using transporters, and those that aren't saturable, that don't use transporters. So the first one is small lipid substances, small molecular, in terms of molecular size or molecular weight, small substances that are lipid soluble can just get into the brain very easily. And we're talking about things, endogenous things like steroids. And most of the drugs that we use that get into the brain and have a CNS effect are small lipid soluble molecules. But again, there's exceptions to that rule. There's two major exceptions. If you're too lipid soluble, you'll get hung up in the membranes, cell membranes, and you won't be able to partition back into the interstitial fluid of the brain. So there's a limit to how, if you're going to build a drug or build a substance to get into the brain, there's a limit to how lipid soluble you want to make it. And another big exception is there's a brain to blood efflux system, brain to blood transport system, that's actually in the cell membrane that faces the blood. And it can catch lipid soluble drugs that are of a particular conformation and spit those out. So that's sort of the exception to the rule. Lipid soluble substances get in, unless they're too lipid soluble, or unless they're a substrate for one of the efflux systems that keep you out. And there's exceptions to the efflux system. It's a regulated system. So if you have inflammation or things like that, you can turn it up, you can turn it off. And there's lots and lots of disease situations where that comes into play and explains how drugs work in patients.
Yeah. So we've got size, we've got fat or water solubility. In general, smaller things will tend to pass through more easily, but certainly not all of them. Small things that tend to be fat soluble are especially likely to diffuse passively through this barrier, but there are exceptions, and it's sort of like an intermediate level of solubility. You have to be fat soluble and small enough to be able to get through the cell membranes, but not so fat soluble that you get stuck in the cell membranes.
Right. Perfectly said.
and so those things that can passively diffuse through, some of them pass through and then that's it, they're in. Other things pass through, but they're immediately pumped back out. The brain presumably really doesn't want those things to be in there.
Well, actually, they don't even, depending on the efflux system, they may not even make it into the brain. So to back up a bit, the capillary bed is one cell layer thick. It's amazing, this barrier, this incredible barrier, is just one cell thick. And so that means that if you were in the bloodstream, you would go into the luminal cell membrane, and then the cytoplasm for the endothelial cell, and then this abluminal membrane, and then you would reach the interstitial fluid of the brain. So the efflux for P-glycoprotein, PGP, is actually the plasma membrane that faces the blood. So it doesn't even get into the cytoplasm of the endothelial cell. So this is really a pretty cool setup, I think.
Yeah, yeah. So depending on size, depending on fat solubility, things might diffuse in. If they diffuse in, they might stay in or they might not. They might
Mhm.
get kicked right back out. And then there's things that kind of get actively sensed and transported in. What do
Mhm.
some of those mechanisms look like? How does the brain decide or sense something that can't passively diffuse in, but needs to be actively shuttled in somehow?
Yeah, there's a whole variety of transporters. There, of course, there's a whole variety of transporters in any cell type. So, and most of these have been discovered by cell biologists not working on the blood-brain barrier. And so we're sort of, I mean, if there's one area I would say of the blood-brain barrier that has needed a lot more attention for decades, it's cell biology of looking at the brain endothelial cell itself. But in general, the transporter systems are for substances that aren't lipid soluble or that you can't get enough in without some extra boost. And the most famous, of course, will be the transporter for glucose into the brain. And it is going at near max, theoretical max. There's so many transporters of GLUT1 as it is that almost every molecule that could be transported from blood into brain is. And that's one reason that we can look at how active a region of the brain is by doing a PET scan or some other scan and looking at blood flow. Because if a brain area becomes active and it needs more, therefore more glucose, you can't do it by upregulating the transporters. You can only do it by increasing the blood flow and therefore making more glucose available through increasing the flow rate. So if you measure flow rate, it's usually very well coupled to metabolic rate in the brain. So then there are others. There's amino acids, and some of those are used in abundance. And they're not going in quite as fast as glucose, but they're often going in maybe a tenth of that rate. So the most desirable ones. And then and again, those are often what we call flow limited. So that if you want more in, you've got to really increase blood flow. Then you get to substances that are transported at a much lower rate, and this tends to be the regulatory substances. They're going in at a very low rate and therefore not affected by blood flow, and so therefore have more of a constancy and can vary which region that they're transported in most by need or something like that. And we're talking about informational molecules like some peptides, cytokines, growth factors, insulin, GI hormones like leptin, ghrelin, things like that.
Mhm.
And in many of these cases, all the work has been done and shown that the rate of entry, the ability to be transported across the blood-brain barrier, has effects on brain function.
Yeah. So various things can get past the blood-brain barrier by different means. They might passively
Mm-hmm.
diffuse in, depending on their
Mm-hmm.
properties. They might be shuttled in through or transported in through some kind of special protein that is specifically designed, so to speak, to get just that thing in, whether it's glucose or amino acids or some other nutrient. You can also have signaling molecules and regulatory factors, peptides, cytokines, hormones, things like this that can also get in. And these things, have their own ways of getting in. They come through different transporters. They can get in at different rates. Something like glucose kind of goes in relatively in abundance and quickly because the brain needs to have some minimum supply of glucose for energy purposes, basically at all times. Amino acids come in a little bit more slowly, and then other things might come in more slowly still. But there's a number of ways that the barrier can allow different things to come through.
Mm-hmm. That's true. Yes.
And so when we think about sort of the basic structure here, so you've got a capillary, a tube with blood flowing through it. It's coming to the brain. Should we imagine a tube that's being surrounded by basically a very tight network of cells that are specialized to be close together so that not just anything can passively diffuse in, and that each contain some pattern of these transporters that will dictate sort of what can get through and how quickly.
Yeah, I think the way to sort of think about it is you imagine a flat endothelial cell, and now we wrap it around so it becomes a tube, and now we stick these tubes together. Where these tubes come together, they're cemented together by tight junctions. That's one of three it's one of three changes that occur, however. So if we go over and look at a capillary bed at, say, the liver, which is probably the leakiest capillary bed in the body, we see two other reasons why there's fluids going across. There's actual sort of holes piercing the endothelial cell proper, and those called fenestrae, from the Latin word windows. And indeed, if you look at an EM, they look like cathedral windows in some cases. And they have little membranes. They come in different sizes. You know, nothing simple in biology. There's if there's one form, there's thousands of forms, it seems like. So there's various plays on the fenestrae. But ultimately, they are sort of holes piercing through the endothelial membrane. And those things can leak through that. And then there's vesicles that form and take things across. And those are missing in the brain endothelial cells. So we have these three modifications that you can only see these, by the way, by electron microscopy. And it's interesting that the idea of a blood-brain barrier originated in about the 1880s, 1890s. And there was a debate well into the 1960s if the blood-brain barrier truly existed as a physical barrier, or if there was some strange thing like the brain cells just couldn't bind dye or something like that. And the reason is because if you look at the capillary grossly or by light microscopic methods, doesn't look like anything special. So people started saying, well, they all look the same. It looks the same as in the liver, you know. Well, maybe the capillary isn't there. Maybe there isn't a real blood-brain barrier. And then in the late 60s, a guy named Reese with colleagues looked by EM, and he found all these changes. And so, okay, there is actually a physical blood-brain barrier. So we were on the moon before we knew for sure if there was a blood-brain barrier or not. I think that's kind of interesting.
Yeah. And so tell us a little bit about the history of this field in terms of what the original ideas are and how those evolved over time as it relates to some of your discoveries. You know, it sort of seemed like in the past, after we established that there was a blood-brain barrier, people often thought it was like a very, very strict barrier, almost nothing got through. And over time, you sort of kept discovering, well, actually, some things can get through, and there's all these different mechanisms of many kinds that regulate this, and it's actually much more complex and dynamic than we thought. What did sort of the general arc of that discovery history look like?
Yeah, most histories will start with Paul Ehrlich, and Paul Ehrlich did some experiments supposedly when he was a student. He was a medical student in Germany, and they had to do a thesis. And so it may be that his thesis is that he injected dyes into animals, and he found that these dyes, there was a group of dyes, aniline dyes, that went everywhere and stained almost every tissue except for the brain. So a deeper reading shows that there were other people doing similar things with sort of different approaches. And so probably Ehrlich gets too much credit. But the reason I think Ehrlich gets so much credit is he later became very famous. He won one of the first Nobel Prizes, and he's considered the father of both pharmaceutical sciences and pharmacology. And famously, he did discover the drug for the first treatment of syphilis. And so he's really a fascinating story. And so to add one more story onto the legend is so tempting. I haven't read this, but I recently thought, why is this guy so interested in dyes? Why is he so interested in staining? And then I think that the answer is this, that he was developing drugs. This was back in the time when the idea of having a pharmacopoeia was sort of new. And he wanted, how do you discover what a drug will do? Well, probably site of action. So if a drug goes to the liver, it's probably going to have effects on the liver. And the only way in his day that you could determine where a drug went really was to see it. Because we didn't have any way, we didn't have mass spec or anything like that to otherwise detect the substance. So I think that his interest in where dyes go was first trial of establishing the principle of site of action. At any rate, so he found that the dyes weren't taken up by the brain, and the great Ehrlich got it wrong. He thought it wasn't taken up by the brain just because the brain tissue could not hold on to the dye. He never thought that there might be a barrier. But some other people were injecting bile acids into the brain and said, look, it doesn't have any effect unless we inject it directly into the brain. Therefore, there's something keeping the bile acids out when we inject them peripherally. And there are some other studies like that. So even though Ehrlich wasn't in favor of blood-brain barrier, there was some thought going on. And one of his students decided to do a simple experiment and inject the dye directly into the brain. And sure enough, the brain tissues took up the dye. So now we're back to a barrier idea. But as I say, it was very difficult to see a physical barrier. And for some reason, people wanted to really see this to be convinced. And so there was a lot of going back and forth in some early studies from a Russian lab, and a little bit later studies from an English lab, a guy named Davson. And they sort of said, well, there's the choroid plexus now also, and it's important, and some things are coming through there. And at one point, it was believed that perhaps that was the only important thing. And the capillary bed was just mechanical, just a barrier. And then Davson showed in the 50s that no, it looks like both are important, and it looks like that the barrier is important, and that it's at the level of the capillary bed. And so then the studies of Rees and Karnofsky and others with the EM showed, yes, there are these modifications there. Then they looked at the choroid plexus. They found tight junctions there as well. So by this time, by early 70s, the blood-brain barrier was established, and it was established that it was these modifications that we've talked about. And then there had already been a lot of work showing the lipid solubility, things crossing, but no one had really looked too much at transporters. There was a little bit of work with electrolytes. And so a guy named Oldendorf came up with a method called BUI. And this allowed him to look at things that are transported across very quickly. And he said, oh, glucose is transported very quickly and by a saturable system, and amino acids are, and those two are different transport systems. And this began the era of looking at transport systems and some of the early tools to do that. And then Patlack, Blasberg, and Finstemacher came up with a mathematical way of looking at substances that cross very slowly. And this allowed us to do our work and look at the ability of informational molecules to cross. So that's sort of the kinetic story. Then there's a more of a cellular story. Astrocytes and pericytes were discovered, and they are the cells that tell the blood-brain barrier to be a barrier. So when they come into the brain, and they do so very early. It was a thought that the astrocyte was the main cell that did this, but now we know the pericyte's at least as important, and actually tells the blood-brain barrier to form in the neonate, in the fetus.
What is this concept of saturable transport? How does that relate to passive diffusion versus active or regulated transport across the barrier?
Yeah. So there's various ways to be transported by a saturable system. So if it's saturable, it says there's a receptor-like protein or a transport protein that's there, as opposed to diffusion by lipid solubility, which is not saturable. So and it comes in sort of two big flavors. Does it require energy? In which case, that's ATP or something like that. or is it non-saturable? It does not require, not requiring energy. That would be a passive diffusion sort of thing. Glucose is actually passive diffusion. It doesn't require energy to take glucose across. Of course, it takes energy to build the transporter and all that, but it doesn't take energy to run the transporter once it's in place. And it's still saturable, but just doesn't require energy. Mm-hmm. And the active transporters are active. Active doesn't mean saturable. Active means it requires energy. It is saturable, but it requires energy. Unfortunately, we sort of the terms get mixed up, and many people who are either novices to the blood-brain barrier or experts but get sloppy. Often, when they talk about transport, they talk about active, but that's only one type of.
Got it. So some substances can basically get through the blood-brain barrier by some means, and all that's really dictating that or limiting that is the concentration in the bloodstream versus in the in the brain, in the nervous system. And some things are going through receptors that can be saturated. So there's this sort of extra regulatory step that is limiting the rate by which the rate at which something goes from the bloodstream into the nervous system.
Right. And those can be energy requiring or not energy requiring. So that would be passive diffusion versus active transport.
Yeah. And then sort of as your career was going and new things were being discovered and debated initially, what were some of the early discoveries around some larger substances that actually do get through the blood-brain barrier, such as peptides, which are basically just multiple amino acids strung together? Are there any interesting examples where maybe there was a debate and some interesting discoveries early on there?
In the 80s and even into the early 90s, there was particularly in America, not so much in Europe, there was a lot of pushback that substances like this could cross. And I'm still mystified why small peptides would have been thought to be too large, because it was pretty much agreed that thyroxine, which is the thyroid hormone that needs to get into the brain, was saturably transported, is about 700 daltons, about 774 daltons. And there are peptides that we were looking at that were smaller than that and able to get in, tripeptides and things like that. And so I think it was just that the only example of a string of amino acids that we had was things like albumin, and we knew that albumin didn't get into the brain. And so I think that it was just sort of an over-application of what little we knew about blood-brain barrier that was overly applied to another string of amino acids, the peptides. But yeah, the first one we looked at was delta sleep-inducing peptide, and it had just been isolated from the cerebrospinal fluid of sleeping cats. And supposedly, if you took this and injected it into the brain of the cats, they went to sleep. I've never seen that. I'd love to see that, because I've had many cats in my life, and anything you can do to control the behavior of a cat is a mystery to me. So that would have been a great thing, I think. But so here was a peptide that we knew was circulating in the blood, occurred in the brain as well, and that had a profound effect on cats, on a behavior. So and it was 9 amino acids long. And as I said before, there were tricks we could do, so we could demonstrate that substance would get in as an intact all 9 amino acids. And so we showed that. And then we had a guy who's a peptide chemist. Very rare in the 80s to have peptide chemists. And this guy was great, David Coy. I think he's still active, and he did fantastic work. And he had made all of these analogs of DSIP, of delta sleep-inducing peptide. And so we were able to label those radioactively, and if you use radioactive materials to look, you have really incredible sensitivity. And these peptides varied in size and composition, and lipid solubility, molecular charge. So we did that thing. We looked to see, what's important here. And what we found is that this class of peptides, they tended to cross. They crossed at different rates. and the best predictor was their lipid solubility. So that said, okay, uh, although they're big, almost 1,000 Daltons, they're still able to use this lipid soluble area and get in at sizable amounts. And so then we said, well, what else can we do? So we went to the catalog and we ordered all of the radioactively labeled peptides that were available commercially then. There were about, I think, 18. And we looked at them with our method, which is in those days was before the math of Patlack. So it was a little crude, but we were able to do that. And we found that a lot of the substances, yes, agreed, the more lipid soluble they were, the better they got in. But there were a set of three that didn't get in very well at all. And we looked at them more carefully and we said, oh, they all have a similar structure. They're less than 5 amino acids and they have an N-terminal tyrosine. And so we speculated that there was a brain to blood efflux transporter for them. And then we set up some methods because there were no methods really to do this kind of work at that point. So we figured out a method to look at that, and we found that yes, there was a brain to blood saturable transporter for small N-terminal tyrosines. And that was an interesting group of peptides because it included the opiate peptide class of the enkephalins. And so that was sort of interesting in that way. So yeah, I can go into the whatever happened after that, but
so some peptides can get in, many peptides can get in. I would imagine here this is an area where we've got some general rules, but there's a lot of exceptions to the rules. You mentioned that the lipid or the fat solubility of the peptides is a major factor. The ones that are more fat soluble generally get in more easily, but there's key exceptions like the ones you just mentioned that have, certain amino acids at one end of the peptide. In general, like, do most peptides get through? Is it really a mix and it depends on the individual peptide? Do smaller ones generally get in more readily than larger ones?
So I would say that most peptides get in, but with the caveat is that besides our lab, there aren't a lot of labs that do this kind of work, and therefore there's lots and lots of peptides that haven't been specifically examined. And of course, we've examined the ones that there's a good bet they're getting in because we know that they have profound effects on CNS. So at least within that group, most peptides get in, and if they don't, there's usually an efflux system for it. So I think one misconception about the blood-brain barrier is that it's all or none. It's either open or it's leaking, or that a substance gets in or doesn't get in. But there's no absolutes in biology. And so it's much better to think about, and you said your audience likes to delve into the weeds, so I'm going to go into the weeds here. It's better to think about the rate at which a substance gets into the brain on a logarithmic scale. So if you're talking about glucose, you're talking about 10 to the minus 6 microliters per gram per minute. That would be the rate. And if you're talking about albumin, you're talking about something like 1 times 10 to the minus 6. So even albumin, a little bit of albumin gets into the brain. It doesn't get in at the capillary, it gets in through another rate, but the point is it accumulates in the brain. And that's why there's a little bit of albumin in the cerebrospinal fluid and some of the things that the choroid plexus does. So that's a millionfold difference. And then there's some things that get in even less than albumin, have rates lower, like IgG molecules. And that's because in addition to being big and not lipid soluble, there's also an efflux system for them. So they get in about 10 to the minus 7.
So instead of asking, does this or that peptide get in or not, perhaps a better way to think about this is, at what rate or to what extent does a given peptide or substance get in? It might be very, very, very slowly and very little, but it's going to be a spectrum of answers depending on the peptide, depending on the molecule, and so forth.
Right. And so the real question should probably be, is the permeability biologically relevant? So, and if it is, under what circumstances? Because it may be important in a disease context, but not normal physiology, for example. So yeah, that's really what it should be. It shouldn't be an absolute, does it get in or not get in? If you have sensitive enough methods, you may very well find it. There's very few substances that we've not found at least a trace, you know? And so it really has to, but we found many substances that trace is like, oh, irrelevant.
Yeah, yeah.
So
So this brings me to another set of questions I have, which is, we can think about a substance's ability to get through the blood-brain barrier, but that itself might change. I want to
yeah.
ask you about how dynamic the barrier is. Can it fine-tune or change how permeable it is to a given substance, both by deciding to do this, so to speak, through mechanisms that are easily to modulate, and also as a function of things like the integrity breaking down over time with age-related decline. So, starting with how dynamic and regulated the healthy blood-brain barrier is, can you start to give us a sense for what that looks like, how easy it is for the brain to play with the permeability to any given substance?
Yeah, I think this is one of the areas that I really enjoyed investigating the most because it does show the blood-brain barrier. Probably we should think about it, first of all, to summarize everything we've done, as an interface rather than a barrier. It's clearly a barrier, but it's a barrier plus.
So the blood-brain interface.
The blood-brain interface, yeah. So I'm not advocating changing 120 years of nomenclature, but it's worth thinking about. This is not just a brick wall. This is really a dynamic area, and so it's not surprising then that many of the key transporters are regulated. And we already talked about how do you regulate glucose because it's going through so fast, and that's regulated by blood flow. But the substances that aren't flow dependent, that get in much slower, they have the ability to be tweaked. And of course, we know that transporters and enzymes in other cell types, and enzymes and transporters have very similar math describing them. And we know those are all, have regulatory sites and all this kind of stuff. So it's logical blood-brain barrier transporters would too. And indeed they do. Leptin, for example, we found which is a substance produced by your fat, secreted from the fat into blood, and you can take a person's blood level, and the leptin level will correlate extremely well with the amount of fat that they have, amount of adipose tissue. And then that crosses the blood-brain barrier and is one of the things that regulate body weight. In a very interesting way. It seems that what it does is when you're starving and leptin levels, and therefore you have less fat, leptin levels drop, that absence of leptin becomes an emergency signal to the brain. You better start looking for food. It's a little bit like your gas gauge on your car. You're driving down the road and it's on full. You got lots of leptin in the blood and in the brain. You don't worry about it. But when you look, oh my gosh, it's almost on empty. It's the absence of that gas that makes you panic. And so it's the absence of the leptin. So,
the absence of leptin is basically telling the brain that the body's out of fat, i.e. stored energy. So we better get some more energy in and store it up. Exactly.
So that turns out to be regulated by a couple of interesting things. One is epinephrine. It tends to increase the transporter, and another one is triglycerides. Triglycerides interfere with the leptin transporter, and that actually makes it, I think, pretty cool evolutionary sense. We think of triglycerides in our modern world as being associated with high-fat diet and obesity, but actually, if you go back to the hunter-gatherer or something like that, triglycerides go up when you start to starve. Because you're not eating, so you're calling on your fat, and your fat will break down to free fatty acids and triglycerides, both of which become elevated in the bloodstream. And so what triglycerides are doing then is preventing the leptin from crossing into the brain and accentuating the lack of leptin in the brain, and therefore the starvation signal. So I think that's an example of a transporter regulation that we can imagine why it's important. And there are other regulatory systems going on, and it's sort of similarly affecting a peptide transport and that kind of thing. Ghrelin's also affected by epinephrine. Insulin probably has a lot of regulators, but we haven't really found those. It varies a lot from experiment to experiment. And so it's probably having a lot of peripheral substances that are affecting that as well.
Mm-hmm. Talk a little bit about how, so some of these things, leptin, ghrelin, these other peptides and hormones, they can cross the blood-brain barrier in a regulated fashion. To what extent is the brain accumulating those things from them crossing the blood-brain barrier versus sampling at the circumventricular organs? Is it some of both? And I guess related to that, when something like ghrelin or leptin or whatever gets through the blood-brain barrier, how far into the brain tissue can it actually go?
Right. Yeah, I think it's going to be integrated, and I think we need to study those integrations much better. Again, the circumventricular organs, again, it's really, remember, it is part of the brain, it's part of brain tissue. Mm-hmm. So there are nerves coming into and out of that and connecting it to other regions of the brain. And yet it's it's connected by nerves to the rest of the brain, but its humoral interaction is delimited by the tanocytes. So again, a rather complicated system that's both sequestered from the brain humorally, but well connected by neuronally. I think that this probably, if I could be imaginative, probably means that a given hormone, such as leptin, can come in through the blood-brain barrier, the choroid plexus, circumventricular organ. All of these rates of entry will be different. The immediacy will be in the circumventricular organ. And so probably that gives the opportunity for an immediate influence on brain function, which may be different from the later or more sustained entry through the choroid plexus or the blood-brain barrier. So those kinds, I think with that kind of theoretical framework, we need to do more work sort of demonstrating that.
Mm-hmm. And you mentioned earlier that there was a structural feature, I think you said a terminal tyrosine that some peptides have. And even though they can maybe get through the blood-brain barrier to some extent, they are effluxed. So they're basically sent right back out. Are there any examples like that of structural features that tend to generally make peptides more or less permeable?
I think that's probably the best example. There's you can also, I think another example is some somatostatin analogs we looked at, but the general principle is that you can make a peptide chain very lipid soluble if you circularize it and make it a cyclic peptide. But those are substrates for the P-glycoprotein system. So it's an example of cheating and making something very lipid soluble, but you get caught up with the PGP, and so that trick doesn't work. PGP itself, it has some peptides as its substrates, also has many other substances as its substrate, is actually a great example of blood-brain barrier regulation of a transporter. And remember, this is efflux, brain to blood. So you got to think in reverse, the enemy of my enemy is my friend kind of thing. But it's, for example, is greatly increased activity with inflammation. One of the things that causes inflammation is seizure activity. And we try to control seizure activity with anti-seizure drugs, most of which are PGP substrates. So one mystery is that when people have status epilepticus, that is repeated seizures over and over and over again, it's really hard to break that seizure cycle. And so as a result, the blood-brain barrier is probably explaining this. The seizure drug is a substrate for PGP, but maybe not a very strong one. So enough gets in to affect seizures. But if the seizure activity sort of breaks through, inflammation, there's more PGP activity, less drug is residing in the brain. So it becomes a catch-22. The more seizures, the more inflammation, the more PGP. So it's really hard to break that. There's a couple of exceptions to that rule. There's a couple of drugs that still work, even in status epilepticus, at least in many cases. And neither of those are substrates for PGP. So the answer to status epilepticus is also sort of dictated in the blood-brain barrier activity. And there's one drug called Akinra, which is an interleukin-1 receptor agonist. And it is useful for controlling seizures in children who have developed seizures because they've had some sort of intercurrent illness and inflammation, and for some reason they get these seizures that nothing seems to break them. And anakinra is able to do that. And we haven't looked at anakinra specifically, but we have looked at the parent compound, interleukin-1 receptor agonist, and it is transported across the blood-brain barrier by the interleukin-1 transporter. And so it makes sense that anakinra is independent of PGP, is able to get into brain by its transporter, and there it's able to tamp down inflammation and treat seizures in these children.
Mm-hmm. When it comes to the dynamic regulation of the blood-brain barrier's permeability, either in general or to specific substances, can you give us a sense for what that looks like in the healthy state? So, for example, things I'm thinking of here, does the permeability of the barrier change according to, say, circadian rhythms or sleep-wake cycles? Does it become more permeable at night when we're sleeping or anything like that? What is the sort of temporal dynamics of how tight the regulation of the permeability is for the blood-brain barrier?
Yeah, I think if you talk about lipid solubility, the membrane composition, which is often the cell membrane permeability to lipid molecules for other tissues is often affected by diet. So if you change your diet in fats or something like that, a cell membrane might become more permeable to something. But in the blood-brain barrier, the brain endothelial cell, that lipid solubility doesn't seem to change. So lipid soluble drugs will get in pretty much the same order and the same magnitude, and say in a neonate versus an adult, that kind of thing. But the transporters are different. The transporters are highly regulated. In the key here to thinking about this is that the blood-brain barrier, and I'm talking now about the blood-brain barrier, and probably it will also include the choroid plexus and any other cell junction that has transporters like the tannosides probably. But the key here is that the barrier or barriers are slave to the needs of the brain. So if the brain's always changing, right? We have a different brain in the neonate; it's growing. We have a different brain in with aging, and so all of these signals that are transmitted to the brain endothelial cell through the pericyte and the astrocyte, but also probably by every other cell in the brain as well. There's all this crosstalk going on between all of these cells. They're telling the blood-brain barrier what the brain needs. And as a result, the blood-brain barrier will adapt and try to give what the brain needs. If that breaks down, that could be a disease state. So let's take, for example, Alzheimer's disease. Alzheimer's has got a lot of blood-brain barrier changes. And we have good ideas, or at least I think interesting hypotheses, we being the field, about what that means. But ultimately, we're not sure. They're still, I would say, at the testable stage. So, for example, glucose transport across the blood-brain barrier of the Alzheimer's disease brain is reduced. Now, we don't know if that is a primary function at the blood-brain barrier. There's decreased ability for some reason for the glucose to be transported across, in which case the poor neurons are starving, and that could be a very big problem in AD, maybe causal. Or is it that the brain cells are less metabolically active to begin with, And so the endothelial cell is simply responding to a decreased need of a sort of sick cell, if you will. We don't know, but we do know that the transporter is not working well. Mm-hmm. We also know one of the hallmarks of Alzheimer's disease is its plaques and tangles. And its plaques consist of amyloid beta peptide. And that's usually transported out of the brain by efflux systems, by other methods as well, degradation and things like that. But about 25% of the A beta is gotten rid of by efflux systems. Two in particular, and neither one of those work in Alzheimer's disease. They go offline for unknown reasons. And it's pretty clear that going offline contributes to the plaque formation. In fact, we did an experiment. We made antisenses against one of them, LRP1. PGP is the other one. LRP1, knocked it out, and A beta levels went up in the brain, and the mouse had all kinds of behavioral learning problems. So those transporters seem to be really critical to controlling the level of A beta in the brain. Mm-hmm. So, yeah.
So, in general, with aging, setting aside individual disease states like Alzheimer's or something else, as we get older, what does the integrity of the blood-brain barrier generally look like as we age? Does it just become more permeable to everything and more leaky? Does it just become dysregulated in terms of its ability to fine-tune exactly what comes in and to what extent? Is it all over the place, or is there sort of a general arc to that?
Yeah, the change in transporters, because you have to look at every transporter individually to talk about that transporter, is a little mystery. We know that there are lots of changes in the blood-brain. Oh, sorry, let me back up. We know that the few transporters that have been looked at, and again, we're talking about the field, not just our lab's work. There are a lot of changes in blood-brain barrier. But in terms of the integrity of the blood-brain barrier, it looks like that there is an increased leakage in the hypothalamus with normal aging. And that occurs around 60. And, I try to be dispassionate about science, but I really hate this fact because I'm over the age of 60. So, to think that my hypothalamus isn't just as good as it used to be really irritates me. But anyway, but here's the thing. Unfortunately, one of the things about BBB, there's a lot of myths, and there's a lot of things that sort of get out and then they sort of take on a life of their own. So if you read a lot of the reviews now, it sounds like the aging brain, even the normal one or the AD brain, either one, it's leaky. It's just open everywhere. And the fact is that the aging brain has a few places where it looks like it's gotten more leaky, but it's not widespread and it's not profound. And here I'm going to go off on a tangent about blood-brain barrier in disruption, because we're getting a huge list of diseases now where there's probably disruptions of the blood-brain barrier. But these are not profound. These do not return the blood-brain barrier to that of a peripheral tissue. Even in cancers of the brain, which are probably much more leaky than anything we see in other states, the blood-brain barrier is still highly restrictive. Albumin might come in 4 times faster, but that's still a far cry from, it's a million times faster slower than glucose. So the barrier is just squeaked open a bit. Now, is that relevant? Probably is, because the brain's very sensitive to disruption and can cause inflammation and go forward. But they're very subtle openings. So so they're hard to detect.
Yeah.
In fact, the MRI detection methods that are used to show these disruptions, they're really at the level of detection mm-hmm. limits. Mm-hmm. And, sometimes, yeah.
And you can imagine too, right, a pathology coming from increasing the barrier's permeability generally, because then it's just more leaky and stuff's getting through that you don't want to. You could also imagine it turning pathological from going in the other direction. If you make it more impermeable, then you can't get stuff that you might want to get into the brain.
Yeah.
Give us a general sense for like what's going on today in terms of how people are thinking about developing therapeutics that relate to modulating blood-brain barrier permeability. So, for example, many small molecule drugs, as we said, can't really cross the blood-brain barrier very well. And my understanding is there are people developing methods and potential approaches where you might say temporarily enhance the permeability of the barrier to allow it to have a drug come in. To what extent is that happening today? And what do you see as sort of the general pros and cons at a high level of playing with the permeability of the barrier so as to allow things like medications to more readily cross it?
Yeah, there's three basic ways to get drugs into the brain or to develop drugs that have taken up maybe 95 or even 99% of the effort. And the first one is production of small lipid soluble drugs, and we've talked a lot about that and its caveats and things like that.
So the basic idea is you've got a small molecule drug, but you're going to make it lipophilic to a degree that makes it likely to just diffuse it.
Yes, it's probably more the other way. We've for decades and decades and decades been able to make small drugs, and those that wound up having effects on the brain in retrospect turned out to be lipid soluble. It's probably serendipity, but now we know that, and so now yes, we can actively engineer small drugs to follow the rules of Lipinski, for example. So that's one way, and that's proven it's right to be there. The other two are disruption of the blood-brain barrier, and then the other one is I'll call as a category is Trojan horse. And Trojan horse is basically anything you have a drug you have a substance that's already crossing the blood-brain barrier, so you just put something on it that will drag it across. And so let's talk about disruption. Oh, and I should also say that although that's taken up those three have taken up the lion's share, there are really a lot of other approaches that we could take. And we wrote a review in Nature Reviews Drug Discovery about 10 years ago, listing all the other approaches that one could have for getting therapeutic effects into the brain other than those three, because the last two, quite frankly, haven't been very successful. So disruption, the idea is that you have the drug in the blood already, but the barrier is there. And so if you open the blood-brain barrier, the drug will go in, and that will be therapeutic effect. And the problems with that is that anything that is many things are able to go in. And even if you make the disruption small, so it's less than 200 Daltons or so, a lot of things that are toxic to the brain, like monoamines, are smaller than that.
Yeah.
So you get a lot of things that are meant to stay out of the blood-brain barrier. Inevitably, this causes inflammation in the brain. Focused ultrasound right now is the biggest area that is being looked at for disruption. Previously, it was chemical agents like antihistamines and things like that regulated tight junctions. And FUS is probably not only opening tight junctions, but also producing increased vesicularization and things like that. And my fear is ultimately it will have the same problem of causing disruption and being nonspecific. But I think, there's a lot of people working on that. So they may figure out how to tweak it, who knows. But I think that's sort of where that field is right now. The Trojan horse field is huge because people have tried all kinds of versions of this for about the last 40 years. And there are promising efforts, particularly in the last 5 years. I think there's been some promising ideas. But it's strange to think that we've been doing this for 40 years and we haven't already solved the problem, because it's so intellectually gratifying. It seems so intellectually elegant. Something's going across, just stick it on and let it get across. So why don't we have lots of shuttles for that? And I think that there's a couple of reasons. So the idea is that you take a molecule that's already crossing, let's say insulin, and you stick something on it, and it plugs into the insulin transporter and is transported across. That's the way it should be. But it doesn't seem like that's what's happening. And I think the reason is because it doesn't route through the receptor any longer. It routes through another pathway called absorptive transcytosis or absorptive endocytosis. And that often gets routed to lysosomes, which break down everything and that kind of thing. And the blood-brain barrier seems very, very good at finding these molecules that have been altered, and saying, well, that's not really insulin, that's something foreign, and routing it over to this different way. And I think that the reason the blood-brain barrier is so good is because there's been, if you go back evolutionarily, as soon as you find an organism that has something that will pass for neuronal tissue and something that will pass for hemolymph, there's something in between that will pass for barrier function. So we have millions of years of the blood-brain barrier being faced with pathogens that are trying to get to that neuronal tissue and eat it up. So it's had to be very clever about figuring out things that are trying to trick their way in. And so we have these millions of years of the blood-brain barrier being faced with substances trying to trick their way into the brain, because there you get this really great meal high in fats and everything, And in fact, some viruses, most of the things that can cross the blood-brain barrier by tricking it are like HIV and rabies virus are able to survive the lysosomal compartment. And so they're probably going to the lysosomes. Most is being recycled back out into the blood, but some is able to get across in the other direction to the abluminal membrane.
Mm-hmm. One thing I want to ask you about, so, if we're thinking about what can and cannot get through the blood-brain barrier and to what extent, when we're thinking about developing novel therapies, one of the approaches is let's try and play with the permeability of the barrier itself to allow something to get through that couldn't get through so well before. Another approach is, well, maybe let's play with something that can naturally get through to a pretty good extent, or modifying something, maybe creating a new peptide using some of the principles that we discussed earlier to engineer something that can get through the blood-brain barrier without changing the integrity of the barrier itself. When we're thinking about those things, we also come to an interesting question in my mind, which is, how do some of these things like peptides that are secreted affect tissues in different ways? So for example, if I've got a gut peptide that's secreted and gets to the brain, or I've got a brain peptide that's secreted and gets to the periphery, do these peptides and other substances generally affect different organs in the same way at the cellular level? Or can they have very distinct effects in, say, neural tissue versus liver tissue or something else?
Yeah, I think one of the reasons there was so much excitement when peptides were discovered is they were what were then called pluripotents. They had all kinds of effects. And they were tended to be sort of mild, and they tended to be sort of integrative. But this was sort of a new way of thinking. And so, yeah, leptin has a lot of effects in the periphery. It has a lot of effects on immune regulation and sort of energy usage in different cells. And I have to admit, I haven't kept up on the leptin peripheral tissue literature, but it's there and it's really interesting. Other peptides, there was one called PACAP, pituitary adenylate cyclase activating polypeptide, PACAP, that was discovered in the 90s, and it was very much in vogue, and it did all kinds of different things. It was a difficult peptide to synthesize and to sort of put in a bottle, sticky and things like that, which sort of held back its progression, but people are still working on this. And I think that the age of peptides, which was envisioned way back in the 80s, is only now coming forward, and we're getting a number of peptide drugs. So these really do have the potential of doing just that, sort of treating a condition or a situation at multiple integrated levels.
Sorry. So you said that this sort of some of this peptide stuff, peptides as therapeutics, that was envisioned back in the 80s.
Mm-hmm.
Why were people envisioning that? What is it about peptides that makes them attractive from a therapeutic standpoint or just a general wellness standpoint?
Yeah, they were sort of first discovered in the 70s, and by the 80s, they were established as being real and not some fragment or phenomenon that wasn't really based in reality. And there was an explosion then because peptides were found everywhere to doing everything and things like that. And there was a big push in the 80s and 90s by some pharmaceutical companies to develop peptides, including for central nervous system treatments. But the blood-brain barrier sort of waylaid those. But they do illustrate a very important point about the blood-brain barrier and how to get around that. And if you want to get a drug into the brain, and one presumes also something secreted in physiological action, might have to think about this too, is that there are really three P's. We've talked about permeability, but there's two others. There's potency, of course, in the brain. If you only need one molecule in the brain to have an effect, or you need 10 molecules in the brain, that potency figures out how much permeability you need. And the other P is the pharmacokinetics. So one of the big problems with peptides and developing them therapeutically is they have very short half-lives, usually minutes. And so if you want to develop this, then you need to figure out a way to make them more stable in blood. And actually, this is true of insulin. Some people consider insulin a peptide, and some people don't, because it winds up having, two parallel chains of amino acids linked together by sulfide bonds. And some purists say, no, you can only have one chain to be a peptide. But at any rate, let's call it a peptide-like substance. We had the same problem. Banting and Best discovered insulin could be used to treat diabetes. And so people started extracting it from pancreas, and this is how Lilly came into existence. But amongst those problems was it had a very short half-life. And so you were often treating, and you didn't know the potency because since it was biologically extracted, some batches would be more potent than others. And so it was really tough. And so insulin had to be stabilized. And eventually, of course, it was synthetically produced, but that wasn't for decades. So it had that's where the zinc insulins and things like this came in. And the peptide field never really went through that. It never really went through being able to produce peptides that were pharmacologically stable. And I'm not quite sure why there wasn't more effort than that. But by making analogs and things like that. But there's, as I say, there's a rebirth of this. In fact, the integrin receptor agonists are peptides. They're derived from glucagon-like peptide 1. And they have this great history of treating diabetes, of treating Ozempic, weight loss, and maybe also affecting neurodegenerative diseases like Alzheimer's and Parkinson's disease.
Mm-hmm.
So,
a lot of these peptides, the GLP-1s and many others, are injected, and so they can have both local and systemic effects. People are also talking about intranasal delivery of peptides. Other drugs are delivered that way sometimes. What does the blood-brain barrier look like in the nasal passage? Is it strong there? Is it relatively weak? Can things pass through the intranasal route that couldn't get through otherwise?
So intranasal delivery to the brain is really interesting. So to save a lot of confusion that I had for about 10 years, let's start off by saying there's three ways of delivering things intranasally. Mm-hmm. One is you've got a sinus condition and you're congested and you just put something there that relieves that. That's a local thing. There's other ways of giving it, and those nasal passages are very vascular, so it can be absorbed directly into the bloodstream. And calcitonin, for example, for the treatment of bone pain is delivered that way. But the final way is you give this very high up at the level of the cribriform plate, and for some reason, substances can then enter the brain. So a bit of anatomy, we have the olfactory bulb and the olfactory nerve, which is a cranial nerve and therefore part of the brain, coming through and going through a part of the skull called the cribriform plate. Looks like a flat plate, and it's got a lot of holes drilled in it. And the nerve goes through there. So you have, as I like to tell my students, you have a piece of your brain dangling out of the top of your nose, which is, I think, a very graphic way of thinking about it. If we can get some substances to that level, those substances will enter the brain. And we understand that in part. We know that there can be retrograde transmission or retrograde absorption, so that substances go up those nerves and into the brain. And there's also the trigeminal nerve, which is in that area. So it can retrograde and go to those regions of the brain. But I think that only explains part of it, because there's some interesting pharmacokinetics that nasal delivery does. And one of them is that whatever that distribution pattern will be in the brain, it achieves that within 5 or 10 minutes, at least in the mouse brain. And we have no way, I think, of explaining how we get that much diffusion, we'll call it, that quickly so fast. So that's a bit of a mystery. And the other is that it can indeed do that. It can distribute all the way from the site of entry all the way to the cerebellum.
So things like peptides, in animals at least, can get pretty deep into the brain from the intranasal route, and that probably can't just be explained by a passive diffusion mechanism.
Yeah, I don't think it can be explained by passive diffusion or any of the other mechanisms that we thought of. Those other mechanisms do explain part of the mystery, just not all of the mystery. So, I mean, that's why science is fun. You know, if all the mysteries were solved, it's time to go to another field. But what this means is that this is really a possibility for treating conditions, and it's been shown to be active in humans. Suzanne Kraft has done some studies giving insulin to the brain. And here's an aside. Everyone, I'm sure, has heard of insulin and its ability to control glucose levels in the blood. But when you get into the brain, insulin has a different life. It doesn't control blood sugar rate of entry into the brain or anything like that. It seems to, though, act as a mnemonic. It has effects on memory and that kind of thing. In fact, every GI peptide discovered, starting with CCK in the 30s to anyone you can imagine, they all have profound effects on brain, on cognition. And memory and that kind of thing. Yeah, it's really. So I can make up an evolutionary theory for that, right, but sort of probably.
Yeah, I mean, I think I could too. It makes a lot of sense intuitively. But so you're saying it's a general observation that a lot of these, what we think of as gut hormones or gut peptides, also have effects in the brain, which affect cognition, memory, things like this. Right. So perhaps it's not so surprising that people are observing brain effects like that for things like the GLP-1s that people are taking for weight loss.
Exactly. I don't think the GLP-1s are an exception to the class, because GLP-1, they're really gut peptides. And but it's been shown for cholecystokinin, somatostatin, insulin, ghrelin. I can't think of a GI peptide that pancreatic polypeptide. They all have been shown to have either behavioral effects or effects on neuronal sprouting and all of these kinds of things.
And of course,
untapped class.
Yeah, it does. I mean, it makes a lot of sense to me. Like, if you just think about an animal in nature, it's going to have to learn and remember and behave in a lot of different ways in order to survive. And what's a big part of survival? Well, it's finding food, it's responding to scarcity, all of that stuff.
Exactly. The GI tract is the first really truly integrated thing, from the first time you see food, so you start salivating and telling the stomach to get ready. Everything all the way to evacuation has to be controlled and monitored, and it's got all these systems. And so it's already a complex system, so why not use that complexity to help teach the brain how to get the next meal? Exactly. I think that's it.
Yeah. So given, the decades of experience you have in this field, studying things like peptides, studying the blood-brain barrier, what do you generally make of the current boom in peptide biology, everything from the GLP-1s that people are using to treat metabolic issues and address obesity, to the research peptides that an increasing number of people are experimenting with themselves. What's most scientifically exciting about this general area to you? And what are some of the biggest gaps you see in the claims and the data that's actually out there?
Yeah, so obviously I'm biased towards brain and that kind of thing, and I'm sure there's all kinds of peripheral tissue stuff that's going to be great, and I don't know anything about it. And so with that caveat of my limitations, I think treating central nervous system diseases are really exciting. And we have a lot. We have everything from depression to dementia. I mean, it just goes on and on and on. Parkinson's, Alzheimer's, of course, are big ones. And I think that this is really where it is, for my interests. And there's lots of things to think about, like how do these really work in the brain, and what do they do, and do we when we start to get down to it, will we need some drugs will affect some types of diseases rather than being universal, and will we need to and other drugs affect others? Will we need multiple drugs? If you look at any big diseases that we have, like hypertension or diabetes, we have multiple classes of diseases, and we often have people on multiple drugs. And so it may be that when we finally get effective drugs for treating neurodegenerative diseases, it may have to be a little bit of a cocktail. We're able to do that for hypertension because we understand hypertension so well, that we're doing salt and water here, and we're doing vasomotor stability there, so we use beta blockers, diuretics, and angiotensin II receptors and things like that. If we get that kind of level of understanding brain function, then we have a really rational way of developing drugs and using them in our patients. But those are probably decades away. Mm-hmm. So more immediately, what we need to think about in terms of the GLP and neurodegeneration is, are all these drugs going to work? Because we have a whole bunch of them now, right? And Ozempic was recently tested and didn't do that well in treating AD. But Ozempic doesn't cross the blood-brain barrier. And so the first question is, how are these substances working? We know that if we treat diabetes, the brain is protected against dementia, but that takes decades. Mm-hmm, These drugs seem to, Exendin and those sorts of things, seem to work rather quickly. So I think they have to get into the brain and work at the GLP or insulin receptors or whatever. And therefore, we need to look at the effect of drugs that can cross the blood-brain barrier very well. Ozempic doesn't cross very well. So I think that we should consider the effect of GLP-1 and IRAs on cognition still untested in the human population.
Yeah. So a lot of these classic gut peptides and hormones actually do affect the brain, they affect cognition, they affect behavior, all of that stuff. Do you think, are people working on other gut-derived hormones other than GLP-1 and thinking about the brain and things like that?
Certainly at the basic science level, certainly in labs. What pharmaceutical companies are doing, though, of course, we don't know. Hopefully they are. Mm-hmm. The problem has always been in science is there can be so much energy and excitement, and things can roll on into the first big disappointment or series of disappointments. People give up. And I always think that as scientists, we sometimes give up a little early in certain areas. Of course, I can also give examples where we get bogged down and we should give up, but we keep on plowing through. But I think that, this is because I lived through this era of peptides. It's just when I was starting. Peptides were hot. Peptides failed as a therapeutic class. And although the decades of discovering all the different peptides and the different things they did continued. So, I hope that we don't get frustrated too early, because none of these things are easy. And they really take an integration of things, particularly if you're looking at blood-brain barrier penetration. A lot of blood-brain barrier is still experimental. It's not theoretical. Yes, we can for example, we can get drugs in by making them small and lipid soluble, except, then we discovered the efflux systems. So it remains an experimental science because we still know so little about blood-brain barrier. And so that makes it harder for a pharmaceutical company to put in the effort to get something past patents and into the clinic.
You mentioned a peptide from history earlier that was kind of intriguing. Delta sleep-inducing peptide, apparently isolated from cats and allegedly had something to do with sleep, although it sounded like that was never proven. Are there any peptides that have a clear effect on sleep and wakefulness or circadian rhythms?
Yes, there's a lot, there's a delta sleep inducing peptide. We looked had a rhythm, circadian rhythm for getting in. Wei Hung Pan about 20 years ago got very involved in chronobiology of the blood-brain barrier and worked with one of the great leading lights of the area and did some work. Not recalling a whole lot about specific examples right now, but it's probably yes that the other thing that seems to happen is not well documented, but that there may be other kinds of entrainments, not necessarily circadian rhythms, but ghrelin, for example, maybe is more permeable near a meal than not, and things like that. That's not really definitely worked out. But usually the changes in blood-brain barrier, again, are relatable to the needs of the brain. And so, in an efficient way. So there's probably a lot of examples like that, but still pretty much unexplored, I think.
Can you talk a little bit more about the timescales on which peptides work compared to other things? So they have a relatively short half-life, usually, on a minutes long scale. So shorter than a lot of drugs that are out there. Can you say a little bit more about that and the extent to which that might be a feature or a bug, or we might want that or not want that from a therapy? Are there any potential benefits or downsides to something that lasts on that type of time scale compared to other therapies?
Yeah, this has, I think, been the major problem of directly adapting them as drugs, is that they're there very, very short time. And the other problem is, of course, that they're by and large not orally absorbed. There's, again, using insulin as an example, there are a lot of studies trying to get in, how can we give insulin other than injecting it with a needle? And so orals were tried to be developed, and those just haven't worked. It's just totally broken down. So there's those issues as well. The action of a peptide, though, interestingly, lasts longer than minutes, particularly if it's given peripherally and you're looking at a brain function.
Yeah.
Some mystery there. And we sort of understand some of the mystery. And again, using insulin as an example, you can almost look at the pathways of insulin. There's the immediate effect. It plugs into the receptor. Whatever it's going to do is done for that tissue, But then it triggers other things, and so there's cytoplasmic events, and those may last for hours. And then it can actually have levels at the nuclear level, altering mRNA production and that kind of thing. And those can last for days. So that actually has been mapped out for insulin. It was a great review in Life Sciences way back when I was a puppy, when I was a student, but I read it, and it was just intriguing how it could explain how these effects, and they're not necessarily effects on glucose anymore. There are other effects on metabolism and things like that. So insulin has a very interesting evolutionary history. It didn't start out as regulating glucose. It was more of a metabolic substance, and had things to do with cell growth and things like that. And its cousin, IGF-1, still has all of that. And interestingly, it seems that insulin in the brain has a lot of IGF-1 effects. And that's why it has effects on neuronal sprouting and things like that. It's got its old metabolic effects, its old effects on cell growth, still intact.
Yeah, yeah. What sorts of things are you working on today, or what do you think is particularly interesting in the field of blood-brain barrier rejuvenation and or peptide biology generally?
Thank you. I'm going to switch over to commercial mode now. There's a lot of things, but there's one thing that I'm really interested in. Recently, Peter Saracen and I wrote a review in Nature Reviews Drug Discovery, and we'd written one previously. I'd written one previously about 10 years previously about all the ways that are being neglected of getting drugs across the blood-brain barrier. So the blood-brain barrier is a therapeutic conduit. But here we looked at the blood-brain barrier as a therapeutic target. And the reason is that since those 10 years, the blood-brain barrier as a site of disease has become increasingly realized. We talked about that there's a lot of diseases that now seem to have blood-brain barrier disruption, but there's a lot of diseases that have blood-brain barrier dysfunctions in the transporters or productions of other things or secretions and this kind of thing. And so there's a lot of basic science and a few other things around that show that we can really auto-target the blood-brain barrier. And again, we talked a bit just passingly about how all the cells are talking. So this is a formal concept called the neurovascular unit. And basically what this does is it takes imagine a slice of the brain, and we become very blood-brain barrier centric or endocentric. And we talk about all the cells that surround that, the neurons, and the microglia, and the pericytes, and the astrocytes, and the mast cells, and everything you can think of. And they're all talking to each other. And so one of the things is this is how the blood-brain barrier knows to be a blood-brain barrier. But it works the other way too. If we could fix the brain endothelial cell, if it's the cause of diseases within the brain, then we're altering that communication. And so one way to treat a neuron might be by first treating the endothelial cell, letting the endothelial cell then affect the neuron. So we have that effect too. But mostly we were interested in diseases that have, sort of a blood-brain barrier story. And so that's sort of my current passion, is to sort of think about that and promote that and work on that. But we're also interested very much in GLP-1. We've looked at about a dozen of them and how well they cross the blood-brain barrier. And it seems that is not predictive of how good they are at regulating glucose, but it is predictive of how good they are at regulating brain functions.
Which of the GLPs cross the blood-brain barrier the most?
One that crosses very well is Exendin, which is one of the very early ones. Unfortunately, it has a short half-life and has to be given multiple times a day, or at least twice a day. There's a group in China led by Horscher who has an analog, and it looks like it crosses very well, and it looks like it has CNS functions. And I'm sure there are other companies as well. But he's recently published on that, so that's how we know about it. So there's interestingly, most of the 12 that we looked at, I think 8, crossed to some degree. And the other 4 crossed very poorly, or even in one case, not at all. So unfortunately, the ones that don't cross at all are the ones with really long half-lives, and therefore very easy to give compared to the other ones. So it will take more effort to do a clinical trial if you have to talk people into giving themselves shots more than once a day.
Got it. So there's a lot of peptides out there for research use that people are buying and experimenting with and trying for various reasons. It sounds like in general, because for a lot of these things, we don't have hard data. We won't literally know for sure if it's getting into the brain or not. But it sounds like in general, a lot of these peptides will cross the blood-brain barrier to some extent. So maybe it's a safe assumption, if anyone out there is experimenting with these things, to just assume that it's probably getting into your brain to some extent for good or for ill.
Yeah, for good or for ill. That's exactly the way I would phrase it. That probably something's getting in. Is it biologically relevant? That should be the real question we're asking. Because on an absolutist scale, and sensitive enough method, you can often find something getting in. Yeah, I don't know if you really want these things in your brain or not. I mean, it depends on what drug you're doing. At one point I ran the hypertension clinics in St. Louis and my VA there, and we had a group that got very interested in herbals. And I was traveling a lot then into Germany and things like this, and there was the ginkgo seed came up for the treatment of AD, which it was well tested in Germany and was found, not to have much. But we talked about how can you do things, in the U.S. And the one person who was very knowledgeable in this area says you can't in America, because if you go and buy echinacea at your local drugstore, the one thing you can be sure it does not contain is echinacea. So I always remembered that, and that, because the FDA doesn't regulate these things, and so you can put anything in a bottle and sell it. And so I really wonder about peptides, which, if you're buying a peptide, they're not easy, they're not hard to make these days, but they're a lot harder than almost anything else. So it'd be much easier to put some powder in there that just, could be whatever, calcium carbonate or something, who would know? So I don't have a trust of non-regulated stuff.
Are there any final thoughts you want to leave people with, or anything you want to reiterate from our discussion today about the blood-brain barrier, about peptide biology, or about any of the topics that we covered?
Yeah, I think maybe that the blood-brain barrier is, without a doubt, the biggest obstacle to drug development. But I think it's not that hard. The amount of drug you need to get in most cases is very, very low. L-DOPA, only about 0.1% of the administered dose gets in the brain. Morphine, if we inject morphine into a mouse, only one molecule in 5,000 gets into the brain. You don't need a lot of stuff in the brain. In fact, you don't want a lot of stuff. There's been examples where people made very permeable substances, and they were just too toxic. For example, morphine its cousin is diacetylated morphine is heroin, gets into the brain like a shot. It's too powerful, So I think that there's a lot of hope if people can sort of step back and do a thoughtful approach to blood-brain barrier. And indeed, in our article that I've mentioned a couple times, is that we try to do that. We try to find examples where you can use the blood-brain barrier to facilitate a therapeutic effect, and things like that. And I think the other thing I'd conclude is that it's a very complicated field, but that's where its beauty is, intellectually, of course. But it's also where its therapeutic potential is, and its ability to do so many physiological things. There's more than one way to skin this cat. There's more than one way in which things fit together. And its complexity and its elegance is also not the barrier to understanding how to use it, but it's the key to understanding all the different ways that we can therapeutically improve drug effects in the brain.
Yeah, I love the term that you used earlier, blood-brain interface. It's not just a cement wall that lets a couple of things through. It really is an interface.
Right. Yeah, it's and very, for lack of a better word, very thoughtfully thought out. And the regulation, particularly by its chief customer, the brain, is able to regulate what it needs. It's a very elegant system, and been a lot of fun studying it for 45 years.
All right. Well, Dr. William Banks, thank you very much for your time.
Thank you, sir. Thank you very much. Been a real pleasure talking with you.
Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio, formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.